I am Dr. Mark Huising. I am a professor of physiology at the University of California, Davis. The research in my lab is focused on the control of insulin secretion from beta cells in the pancreas. Insulin is a signal that communicates to the rest of the body when we are eating a meal. It is absolutely essential. Ask any one of the roughly 1.5 million Americans with autoimmune diabetes who no longer have beta cells. Without synthetic recombinant insulin made possible by science, they would die. Millions more Americans suffer from type 2 diabetes. Their beta cells still make some insulin, but the cells in the rest of their body have gone deaf to the message that insulin is trying to send to move nutrients such as sugar from the blood into storage for later. As a consequence, blood sugar levels of patients would stay too high for too long, leading to all kinds of bad complications. Fortunately, there are a number of medications we can prescribe to patients to help them manage their diabetes. Many people will have heard of Ozempic, Wegovy, and Mounjaro. These are drugs that instruct beta cells to release more insulin. But as scientists are now uncovering, they also help to reduce our addiction to foods, resulting in weight loss. This weight loss directly improves the ability of cells in our body to listen to insulin again, which is a major win-win. What not many people may know is that this entire class of drugs originated with Exendin-4, a peptide discovered in the saliva of the Gila monster, a venomous lizard. Exendin-4 was discovered by Dr. John Eng, who noted the similarity of this peptide to GLP-1, which is our own hormone that Ozempic is based on. He thought it could be used as a diabetes drug, but the Department of Veterans Affairs, who employed him, declined to pursue a patent. So Dr. Eng filed the patent himself and spent several years trying to get the big pharmaceutical companies to turn his discovery into a diabetes drug. They all declined until employees at a smaller company based in San Diego recognized the potential of Exendin-4 and ran all the necessary early clinical trials, which looked very promising. They partnered with a large pharmaceutical company, and now they were finally interested, to collect all the necessary data to gain approval from the Food and Drug Administration 20 years ago. So, the serendipitous discovery from the saliva of a lizard started a whole new class of drugs that was valued at well over $53 billion worldwide in 2024 and is helping untold millions manage their diabetes, weight, and possibly addictive behaviors. My lab is funded by the NIH. I am the PI on 2 federal grants that focus on signals that regulate insulin secretion. Several of my trainees also receive fellowship support from the NIH. Currently, this funding supports 9 trainees on paid positions and provides invaluable research experiences to dozens of undergraduate students. If this funding is reduced or lost, the research done by members of my lab would stop. I would likely lose unique research tools that are impossible to replace. Trainees in my lab are already reconsidering their career path, and incoming graduate students are struggling to find a lab that can afford to hire them. This situation is already stymieing the vital trainee pipeline that US biotech and pharmaceutical companies rely on to maintain the edge in their internationally competitive fields.